So hello everyone. Welcome to another episode of our conversations with scientists from all over the world. Our guest today is Michael Crone. Michael is currently a research assistant at the Fremont Group working on the development of high throughput diagnostics in Imperial College London. He is currently supporting the Imperial College Lighthouse and Northwest London Pathology SARS-CoV-2 testing laboratories and the automation lead. He is also part of the UK DRI Care Research and Technology Center, focusing on developing point-of-care diagnostics. Apart from these projects, he regularly collaborates and mentors students from the Royal College of Art and organizes workshops for innovation design, engineering, XY module. So I was really interested when Michael mentioned that about open source SARS-CoV-2 variant identification. Yeah, so let's welcome Michael. Hello, Michael.
Hi. So I think it's the variant identification is quite interesting, and I'll probably say a few controversial things about this. I mean, I've seen like a lot of people obviously working on using the Arctic protocol for doing variant surveillance, but it really depends what you're trying to do. And I suppose this is a good opportunity to ask you how you see variant surveillance as being important from a public health perspective or from a clinical diagnostics perspective, because often, I mean, the turnaround times, even in the UK, 48 hours is too long in order to make a clinical decision. You wouldn't be able to say, like, this person has a variant, so we want to treat them differently. They need to go through a different pathway. So, but obviously from a surveillance perspective, the goals are different. If you are trying to make sure that you can pick up certain variants, new variants, then you have to be doing whole genome sequencing. But if you're trying to pick up other well-characterized variants, you don't have to do whole genome sequencing, I suppose that's kind of where I'm coming from. So, I mean, I can talk a bit about the work that we've done and kind of where I'm going next and why Khalil kind of, why I approached him, because we're planning to share some of the materials that I've made, but then we can discuss, I suppose, around that exactly how you see genomics playing a part. Because I would even argue like COG UK in the UK hasn't really been used to make public health decisions. So they spent a lot of money, sequenced a lot of things which have been very scientifically interesting, but in terms of actually making a public health impact, I'm not sure whether there's anything that you can kind of point to and say, this is as a result of COG UK. Okay, so let me quickly share my screen. Okay, so I'll just briefly talk about, so this is me, the kind of some of the stuff that we've done so far and our kind of COVID work around this. So what this project started off as is that we come from a kind of a normal lab at a university. And so a lot of it was around building credibility in the first place, because you can't just go to a clinical lab and say, we would like to help you with testing without any sort of credentials or anything around that. So what we tried to focus on was kind of developing some of these standards for diagnostic assay development, because working with SARS-CoV-2 in the UK was quite difficult because of the biosafety level requirements, because it was initially, well, it is biosafety level 3 for research purposes and biosafety level 2 for diagnostics purposes. So an ordinary lab doesn't necessarily have the infrastructure required to handle those kinds of samples. And so exactly, these are kind of the things. And you can just use kind of RNA standards from Twist or another company, but they don't really reflect kind of the full sample processing steps that you need to go through. So the extraction and then the kind of amplification for detection. So we've tried to use these MS2 virus-like particles as standards, and you can package them with any DNA or RNA sequence. They're stable when you spike them into blood or saliva or VTM, and they don't have any special biosafety requirements. And they're quite easy and inexpensive to manufacture. And as it's a protein shell, it kind of protects the packaged nucleic acid from nucleases and other things. So as I said, they're quite easy to work with. So we started off by looking at the N gene of SARS-CoV-2, because this was what the CDC had initially recommended for their primer sites. And I expressed these, purified them, so just using affinity chromatography, And then you can also use dynamic light scattering to get an idea of you get these monodispersed particles. And they look exactly like essentially this image in the bottom right-hand corner. This is a cryo-EM, or this is a structure based on the cryo-EM of these particular particles. And once we had kind of initially characterized them, we wanted to quantify them because that's what you need for a standard. So we did digital PCR as well as qPCR using the CDC primers to quantify these particles. We also then used them, we spiked them into reactions and tested the 3 different CDC primer sets, so N1, N2, and N3. And we tested 3 different qPCR master mixes. And the idea of this was trying to build up a resilience because there were lots of supply chain issues at the start of the pandemic. So we wanted to make sure that you could use any qPCR mix that you wanted to, and they all behaved roughly in the same way. And so these particles have now been kind of allocated an international unit per mil. So we've submitted these as part of this study called the Coronavirus Standards Harmonization Study. So our kind of particle, together with 7 other commercial particles, has been given an international unit equivalent. So it's comparing it to the WHO standard of SARS-CoV-2. So I would like to say that they're reasonably credible as a standard. And it's also it's a nice approach because it's kind of a completely open source. They're off patent, so everyone can make these particles. And so once we had developed these particles, we then decided to use them to make a reagent agnostic high throughput diagnostic workflow. And the idea for this was that at the beginning of the pandemic, a lot of hospitals were very dependent on Roche and Abbott machines, and they couldn't scale the supply chain. And so the problem was you had shortages and it was very, very difficult to set up diagnostics. So we wanted to set up an automated platform where you could use different plastic consumables, different kind of RNA extraction kits, and scale accordingly based on that. So we kind of used the equipment that we had in the lab to kind of develop this workflow. So we have these liquid handlers called SciBioFelixes, which kind of tapped a slightly different supply chain to what was being used by a lot of the other companies. And so you could get extraction in 62 minutes of a 96-well plate and PCR in another hour. So we kind of extrapolated that in 2.5 hours you would have kind of samples through this entire workflow. And so we initially used our virus-like particles to validate these kits, or to validate this entire workflow. And we showed that you could get low intra-run and inter-run variability and comparable performance between the 2 kits that we tested. And we have validated a few more, but we haven't had supply challenges since the beginning of the pandemic. And we showed sensitive detection to below 1,000 copies per mil. And these results then translated quite well into kind of a patient sample validation. So on the left-hand side, we had us, which is LBF, the London Biofoundry, compared to Northwest London Pathology, who were using a completely different testing platform. And we showed really good correlation between the 2 results. And we also showed good correlation between the 2 different kits that we had kind of validated on these platforms. So that's kind of the work that we published, but I suppose the work for us didn't really stop there. So we developed a testing workflow in collaboration with our NHS colleagues, which has formed the basis of patient and staff testing during the pandemic there. We also developed a testing workflow for Imperial College, the Lighthouse Lab, which was essentially part of the track and trace in the UK until the end of March. And they're currently using this lab for staff and student testing at the university. And altogether, now it's probably closer to 500,000 tests to be done using what we developed. And these are just some of the images of the lab that we set up. So really nice biosafety cabinets. And then kind of the automation platforms. So I suppose more relevant to this discussion is then developing an assay for clinically actionable variant detection. So as I said earlier, like whole genome sequencing is too slow for clinically actionable results. You can learn a few things around infection control and things like that, but often, or you could potentially make public health decisions, but the timescales are a lot longer than what would be required for kind of a clinically actionable result. And kind of the situation that I think is that if you have a specific variant that might have vaccine escape potential, you might want to quarantine that person and put them in a slightly different ward or different hospital potentially, and you need to make those decisions quite quickly. So you can do Sanger sequencing, and that's what I'll be talking about, but this is potentially also too slow. So we're focusing on a slightly different approach. But I think it's just kind of bringing together the topic about if currently maybe we don't have to make decisions based on variant detection, but if we did want to, how long would it need to take for it to be useful, and what would we need to know? So something that I've done, and why Khalil contacted me, was essentially I have a library of virus-like particles with the mutations of the different WHO classified variants, and I've added 2 more onto this. So I now added on mu and the new South African C1.2, I think it is, variant, which hasn't yet been given a WHO name. And the idea is that, okay, so these are covering a certain portion of the spike protein. I can look at the exact amino acid numbers, but I think it's 322 to 800 and something. And if you just cover that region with Sanger sequencing, you can differentiate between all the different WHO variants. So these are all the mutations for the different variants, and these are the WHO variant names, and you can see which mutations are present in which variants essentially. And so now we're working with some colleagues, because this becomes a math problem, to try and to work out what are the detections that what are the mutations that you need to detect. So if you were to design an assay that only detected a specific mutation, which ones, like what is the fewest number of tests that you would need to do to be able to pick up and differentiate between all the different variants? So that's kind of what I'm moving on to next. But I suppose the start of this was just to kind of create this library of different virus-like particles that we can distribute to other people who would find it useful to kind of develop their own diagnostics based on that, based on this platform. So I suppose that's kind of the start, and then as it's basically just to kind of have a conversation around that and see exactly what you would find useful. So I mean, we are making these constructs freely available, and we will probably, I still have to make the actual virus-like particles. So at the moment, they're just the plasmids that have kind of the target site, and you can express them yourself and give them, yeah, you can make the particles yourself. But I think basically the start of this was just to make the materials available for everyone under the open material transfer agreements, so that people can also have companies that then can use them to make diagnostics.
Cool. Thanks so much for that, Michael. I'm curious. I mean, I'm the least technical guy here in this conversation. I have a bit of background in biology, but like, how does this work? Like, how is this, particularly the MS2 virus-like particle? I mean, and how does it? How would you explain to a non-technical person how this detects a variant, or at least first the diagnostics? I guess that's the first step. How does it detect that this is there's SARS-CoV-2, and then how does it differentiate the different variants?
Well, so for us, it was just a standard. So what it basically allows you to do is you can package a certain part of the coronavirus genome into a particle, and then it behaves as if it's the virus. So any diagnostic that you then develop for that particular region of the genome that you package inside, it's kind of, the material is kind of relevant for that development of that diagnostic. So it's in the same way. I think the problem with developing diagnostics for variants is that you can't get the virus that has that particular variant. Like even in the UK, we work quite closely with some of the labs that are characterizing like mu and some of the newer variants. But to get your hands on those variants is quite difficult. And so the idea is that you want to have, like, let's say someone in the Philippines picks up a new variant. It'll take a while for you to share that variant with other people, let's say in the UK or the US, to culture it and maybe distribute it to other people. So what you want before that is you want to have kind of like maybe a virus-like particle that has some of the same mutations, so that you can share that material around the world, and you can start developing diagnostics so that you don't have to wait until you get the variant in your country when it's too much of a problem to actually deal with. That's kind of the idea around. So even for the variant diagnostics side, I think the start for me was basically having this library of these particles that have all the mutations, because now you can say, okay, Sanger sequencing, you can PCR out that specific region, do Sanger sequencing, and diagnose a variant based on that. And the idea is that you can have this workflow then up and running using these standard materials before you move to your clinical samples, when it's more expensive and there's kind of limited clinical samples, so you don't really want to do it first on those samples. You want to first do it on like a standard and then move on from there. Because I would say so it's a Danish group that made the Sanger sequencing workflow, the protocol that I shared with you. They're quite supportive. Like if you ask questions on their GitHub, they get back to you and things like that. But they have set up a protocol to sequence across that region and pick up all the mutations.
So
and then
this
you could say this is this particular variant.
You're creating like a practice, what you call this, object where you could design and test diagnostics operations and what you call this, variant identification or the operations without exposing yourself to the virus, and it's also more accessible.
Yeah, exactly. And obviously, like this is the first step for me, and I'm going to look at like a slightly different diagnostic approach for picking up those variations. So it would be different to qPCR, for example. But as you say, like the thing for me at the beginning was just make sure that all the materials are available and that other people can start developing things around those materials.
Amish, you have a question?
Yes, thank you, Michael. Really very interesting talk. I currently head the diagnostics team at PGC Mindanao, so this kind of information is very important for us. I have a few questions. The first one is, in terms of cost, what would be a better alternative? The VLPs are using gene blacks, because right now I've tried using gene blacks as controls. They work just fine, although the limitation is it's not recommended to use them for PCR applications if you target a region more than 1 kb. But since for diagnostics, you usually just target a short fragment, like around 500 or even lower than that for real-time PCR. So it might that might also work for us. So in terms of the cost and maybe infrastructure requirements in order to generate the VLPs, I'm just not sure what would be like more feasible for, like, let's say a low resource lab like us.
So the first advantage of VLPs is that it's RNA. So you have you don't just need PCR, you would need RT-PCR. So it kind of mimics the workflow that you would need for actual diagnostic workflows. In terms of cost, I need to work out exactly how much. Do you do protein purification? Does anyone in your lab do protein purification?
I don't have any experience for protein purification yet. Ate Joan, have you done that before?
No, not yet.
Yeah, so
it would be relatively straightforward to do the protein purification, because it's literally you express you transform the plasmid into a strain that produces the protein, you induce it, and then all that you need to do is kind of break open the cells and use a nickel to bind to the particles and do a few washes. So as part of my goal is actually to work out what is the cheapest way that you could do this. But we do plan to also make them. So I will make them myself and try and distribute them as well. But those are kind of some of the advantages. So and it's also like, even if you weren't to make the particles, you would still have the plasmids themselves, which is then the same as having a G block. So you can then grow up the bacteria, mini prep them again, and then you have kind of an endless supply of G block, I suppose.
Yeah, because we've had the same problem for our ASFB diagnostics. When the outbreak started, we did not have access to validated clinical samples. So we kind of had some problems running out of control. So our assays becoming validated. We had to repeat runs. We have we wasted a lot of resources. And we also had some hurdles during our accreditation because we couldn't generate a stable supply of controls. But we've tried to find a way, but we've had to exercise rigorous what do you call this QC and QA to convert our clinical samples into reliable controls. So we've worked with that, but using VLPs would really make our lab more readily prepared to provide service for, let's say, newly emerging infectious diseases. In that way, it will be it's much easier to synthesize VLPs than to get access to clinical samples from, let's say, index cases. My second question would be more on the technical details and how you would use. So what platform are you planning to? Are you going for RT-qPCR for the discrimination of the variants for that region? Because right now I'm interested in your approach. Like, how will you or will you be doing a multiplex, or are you planning to I don't know, any modifications that you might want to employ to strategically I'm not sure if it's possible, like you just have one reaction and then from the qPCR profile, are you able to detect the specific variant? I'm not sure if that's possible.
I mean, you could do melt curves, but that's quite difficult, I think. So that's what some companies melt curves. So that's what some companies do with CyberGreen. You can melt and then you get a different profile depending on whether there's a mutation or not, because it changes the melting profile. I'm planning to use CRISPR. But I mean, this would not be a clinical, the idea would not be to translate it directly into clinical applications. It would just be to see whether it's possible.
I think melt curve would be very challenging because for instance, for many relative beta coronaviruses, the differences in melt curve is quite small. And I've tried doing universal detection for beta coronaviruses, and sometimes it's difficult to interpret the CyberGreen profiles. Well, I use GeneBlack, so I am not sure if I would have gotten better results if I used VLPs. But when I use GeneBlacks,
I mean, it should be the
really
same.
had to. It's quite difficult to discriminate the profiles. So, like for instance, the differences in the melt curve temperature, the profiles, and sometimes other factors would produce multiple melt curves, even if you only have a single band.
And the problem is you don't want something where you have to sit in front of the qPCR machine and work out whether it's actually there or not based on the melt curve. You want something that's either yes or no. Otherwise, it just makes it very difficult to actually roll out as a diagnostic.
Yeah. So I think so far that's my question. So it's interesting that you are planning to use CRISPR. So, but we don't have that yet here. So hopefully in the near future we'll be able to establish CRISPR workflows in our lab as well.
Joanne
so maybe
and Paul
and Joan has a question.
I just have one question, and it might not be that it might not be that relevant to this talk. But I thank you, Michael, for that interesting topic. I'd just like to ask, because to be quite honest, I'm not very updated right now, because you mentioned Sanger sequencing for variant identification. So I think it's just doing PCR on the spike region. Is that correct? Like what you mentioned, like the 300 to 800 amino acid position of the spike, just so you could because those are where the relevant mutations of the spike, right? Like for identifying variants. Is that correct? Is it?
Yeah, exactly. So I mean, obviously, like whole genome sequencing is overkill to a large extent because a lot of the information that you get is useless. It's like it really depends on why you want to say that there's a variant there. And yeah, I understand, like from a research perspective, being able to track the lineage or to track like the phylogenetics of a particular outbreak might be interesting. But from a, is this a variant? Do we have to worry kind of question? It's much cheaper to do Sanger sequencing and you get the same answer and much faster. Because I assume that you, like in the capital or even like where you are, you probably have Sanger sequencing workflows, because that's like, I'm making an assumption here, but I mean, that is my assumption that it's quite easy to do Sanger sequencing, not necessarily so easy to do next generation sequencing.
Yeah, we don't have Sanger sequence. We don't have a Sanger sequencer actually for one here in PGC Mindanao. But I think there is like in PGC Main, but not in PGC Mindanao. So I think my question is just, so is this like a routine thing already? Just doing the PCR of that 300 to 800 amino acid position in the spike protein, just so you could do variant identification. So it's like
Yes.
validated, or like it's already,
So as far as I know, the
it's like good
protocol
enough.
that
Yeah.
the protocol that they do in Denmark that they have on protocols.io is like what they were doing before they were doing NGS, and they use the Arctic primer set. So you basically just use the flanking Arctic primers that target that region, and then you just do Sanger sequencing of that region. So I mean,
or maybe like you can also use the nanopore, I think, more high throughput. Is that,
yeah, exactly.
is that what they're also doing? Yeah, rather than doing Sanger sequencing. I don't know.
You could do that, and you could make it. You could do like 500 samples together. You just need the barcodes,
Right.
because you don't need to have the coverage that you need for the whole genome. But you can kind of order those primer sequences with unique barcodes on them from the beginning.
Are they doing that? Like
So.
is that like a routine? What? Sorry.
I don't think so, because a lot of these things are like,
okay.
what would you do if you wanted to decrease cost? Like in the UK, you just sequence everything because they don't even consider Sanger sequencing because cost is not an issue. But I mean, like Sanger sequencing probably costs £6-7 a sample, maybe $8-9 a sample. So it's much cheaper than going the Arctic route. And again, you get a result within like 24 hours. You'd have to see obviously where you have a Sanger sequencer, but like the results that we get are within 24 hours.
Okay, thanks.
Yeah, I think this is very important, especially in the point of care diagnostics. So, like for instance, but I'm also interested if you plan also to like extend the scope, like maybe in the detection of co-infections, because right now there's very limited tool to be able to detect co-infections at a relatively fast rate. Maybe that would be very relevant if you want to like manage differentially manage a particular patient. Because so far, what I saw in the literature is mostly looking at the read abundance, especially for sub-consensus variants that might be indicative of co-infections. But there are also some papers which did also Sanger sequencing to detect co-infections. But still, that could be one application of the workflow that you are trying to develop.
And I mean, you could try that with the VLPs, where you kind of like spike in different concentrations and see what you get when you sequence it, for example, to kind of mimic that kind of co-infections. But I think I kind of try to challenge the thinking around a lot of these things, because I think a lot of people do things because they can, or because it's like the research angle is interesting, rather than what would be the best approach for answering a specific question. Because I don't think they're necessarily the same thing, especially around the NGS kind of thing. I think you can answer the same questions and get the information a lot cheaper, a lot faster, using different approaches, like Sanger sequencing.
Very interesting, CRISPR diagnostics. Like, could you explain? I'm not familiar with how CRISPR diagnostics work. Like,
so there's kind of basically you design a guide RNA that detects the single base pair mutation, and then when it detects that, it also cuts it kind of activates the protein and starts cutting other sequences in the in like a solution. So what you generally have is a fluorophore quencher, kind of like what you have for qPCR, and it then cuts that, and then you get fluorescence as the output. So you can't do lots of things together. You would have to have like spatially multiplexed, so one reaction per variant that you want to test. But it's quite low cost. And I mean, the idea would be you kind of start off with PCR of that fragment, and you just take 1 microliter of the PCR and put it into each multiplexed kind of variant detection that you want to do using your CRISPR-based detection.
How far is this in the future, Michael?
I just have to do it. No, I mean, it's not like this is very, very novel. I think like people have done CRISPR diagnostics before, so it's just expanding on that.
I was like,
I think
how long do you think this will be a standard? Like something we could just pick up as like an existing thing and then apply here in our location?
I mean, I think everything will start to move more towards point of care diagnostics. But the problem is PCR will eventually become so good and so fast that I think, and because it's so sensitive already, it's quite difficult for other technologies to compete. It's more like building a device that can actually do PCR quickly at the point of care. Because I mean, you have things like GeneXpert and things like that, which work really, really well and are relatively cheap. So it's quite difficult to see like LAMP-based diagnostics or something else taking the place of those systems, unless there's a very specific reason why you can't do that.
Okay, and also
I think many labs are also we have some labs have been doing a GeneXpert, but their main problem is really in terms of throughput and the cost, because I think it's a closed system, if I'm not mistaken. It's quite challenging for them to source out materials.
Yeah, but again, it's like you need to ask the question, when would you need high throughput diagnostics at the point of care? They're probably a kind of a relatively limited number of applications. And again, it was an interesting question because like in South Africa, they do, obviously HIV is quite prevalent. And so they do like something like 7 million HIV viral loads a year. And you kind of ask the question from a public health perspective, because you can view it at like, you could stratify it at different levels. You can say from a clinician's perspective, they want a result immediately so that they can make a decision. But then if you ask someone from the public health perspective, they say, whether I know the result today or I know the results in 2 weeks, it doesn't change the public health impact on the patient. And in fact, if you're doing something like PCR, where it takes an hour and a half for the results, the patient doesn't want to wait an hour and a half. So they're only going to come back 2-3 weeks later anyway, because they don't want to take the time off work. And so a lot of these things, when you're thinking about like a point of care diagnostic, come into play, because if you can't give a result very, very quickly, people don't care. It's like, it doesn't change. And if it doesn't change the public health impact, it's like, we all can say that we want point of care, but if it doesn't have any impact on the public health problem, then why are we doing it? When a lot of centralized testing can be done that's not as expensive and is high quality, because one of the problems will be like, if you have point of care diagnostics, doing the QA and QC is not necessarily so straightforward. Whereas if you have one centralized lab that's doing 1 million samples a year, for example, you can do a lot of that stuff in-house and do it properly and not have to worry about all these kind of satellite sites.
Yeah, Michael, I'm involved right now in this iGEM team, which is trying to create a frugal biofoundry, and it has some like testing out some approaches on how to do that. For instance, instead of using E. coli, they plan to use like Bacillus subtilis and Pichia pastoris, so that they avoid the purification step. I mean, the equipment to do that is still expensive. Could you share like how you plan to like make it more? You mentioned this, like you're planning to do something like that as well, to make it more frugal, more accessible.
Well, I want to see, because like we have the fancy equipment, so it's often not a good comparison. So I want to see how cheap you can actually make it if I don't use the fancy equipment. But a secretory system might be more complex because you kind of rely on because you like produce the protein, it basically has this like coat protein that packages itself around the RNA. And so my worry would be is that it needs to be protecting the RNA before it's secreted, and you probably can't control that. So I don't know whether it would work very well in kind of a secretory system. You might just like degrade the RNA, so you have the particle outside assembled, but it's not very helpful if there's nothing inside. So I don't yet know about that. But yeah, I mean, I'm quite happy to share the materials, and people are then willing to try, like, are welcome to try it. But I haven't done like peakier expression before. So maybe out of the current scope of what I'm doing. But yeah, I mean, I'll see how low cost you can make it just with the normal traditional approach, and see. I mean, it also depends what's reasonable. It's like, I think reasonably now, you could probably do it for like $100, and you would have enough material for like millions of reactions. Because I think when I made the SARS-CoV-2 reaction, for example, it was like I made the protein, I diluted it like at least 10,000 times. It might have been like 100,000 times. And that was like 1,000 copies per microliter, which is still like a reasonably high concentration. So like you have loads of particles there from a single purification.
Is this doable in like most public or like open biofoundries? Like for instance, if we couldn't do it here in the Philippines, maybe someone in Singapore could do it for us, and then they send us it or something like that.
Yeah. So you mean the biofoundry at NUS in Singapore?
No, maybe that's probably the nearest to us, which could which have the same level of equipment that you do.
Oh yeah. So I mean, we've worked with the foundry in Singapore, for example. We shared the VLPs with them actually. And so have you heard of Jenny Malloy? So she does a lot of the, yeah, exactly. So I mean, essentially I'm now I'm sending the VLPs to her. I mean, just the plasmid constructs later this week, and she's going to send them around to other labs. I think she's sending a few to Ethiopia and places like that. But yes, I mean, the challenge with a lot of these things is I need to test like the cold chain requirements, because in theory they should be quite stable at room temperature for a certain period of time. And obviously that's the limitation with these, is like you can ship them on dry ice, but for a lot of places that makes it very, very complicated. So you want it to be kind of ideally not shipped on dry ice, or maybe refrigerated or like on wet ice, or I don't know, with the blue ice or whatever they say it is the best, the next best option. But yeah, that's kind of like drives the kind of looking at a frugal approach to actually purifying them, and what is acceptable in terms of the cost of purification. Because the problem also then becomes the quantification of the particles. And so what you might find that you need to do is actually have a kind of a constant sequence that you always put into the particles, and then the people just do a qPCR. And so you have like a centralized, like our lab, for example, would do the digital PCR on the particles to quantify them. And we would then do like a qPCR equivalent of those particles. And then if you follow the same protocol using qPCR in your lab, you can then correlate that to the quantification the value that we have. And you don't have to do digital PCR to quantify the particles. So that would kind of be the approach that I need to, again, look into in the future. But for example, yeah.
That would mean that we don't need to do the CRISPR-Cas and the LAMP approaches anymore. So that's the qPCR already.
Yeah, I mean, this would be for quantification of the particles to kind of use as a standard.
Mm-hmm.
But I still think like I'm still quite a fan of doing Sanger sequencing for the variant detection. But that's a very personal kind of thing. I think a lot of people haven't really looked at that route, and there's obviously, even from your perspective, there's a lot of pressure to look at MNION sequencing in the Arctic protocol, but it is quite hands-on and quite expensive.
Yeah, because at this moment our lab is capable for RT qPCR only.
Yeah, no, exactly.
CRISPR-Cas or LAMP.
Yeah.
Cool. Any other questions?
So far, I'm good. It was really interesting.
Cool. All right.
Hey, how much is it costing you now to do the Arctic sequence? I kind of tried to get an idea of how expensive it is to do sequencing using MinION, but didn't really get a good idea.
That depends on how many samples you process together. You can obviously reduce the cost if we process more samples. But again, we're faced with limitations in terms of the number of samples how many barcodes we have right now. Nanopore has a 96 barcode kit, but a team from Denmark was able to modify that, and they were able to get 4x96. They kind of modify the primers themselves. Yeah, I think it's still relatively cheaper compared to Illumina and other sequencing platforms. And the cost drastically decreases if you multiplex maybe at least 48 samples per batch.
So how much the cost to do 48 samples?
Wait, let me check. We had a costing.
So it's like 500 for flow cell.
Yeah.
And then for one, what you call this, kit, an EB kit for the RT protocol, it's like also around, what was that, $500 as well. So, and then the barcodes, the thing is for multiple, how many uses, how many like sets of sequencing is that, Mitch?
Sorry.
The barcodes, the 96
if
barcodes.
you have the 96 barcode kit, that's good for like 1,440 reactions. It's around if we process 24 samples, the cost per sample is estimated to be around how many is this? $40. How much is that? 40 times? Did I get my math correctly? So it's like around 10,000 plus pesos. If we do 48, we can reduce this up to 80% of that, so around 8,300 pesos. So we did like a thorough computation for the costing.
No, that's $400, Mitch, and then it becomes like $300, or no, $200. 8,000 is $200, some around that number, no? So
but that's like the overall cost if you factor in everything: consumables, utilities, manpower.
that's already okay. Just for consumables, when we computed it before, just consumables, it was around $60 to $70 with like 48, or sorry, 96 per flow cell. They just released actually a variant of or a version of RT protocol, the Midnight protocol, and the whole package. I mean, and it seems like it's only $11 per sample. So, and you know, they just package it, everything together. So yeah, that's the thing really is, I mean, it turns out what we learned from the project is actually, it's not really the, you know, it's not the bottleneck. It's not the actual materials or the technique or whatever, or the machine. It's actually getting the reagents here, because like,
exactly,
you know, it's like, first it's they get damaged because they spend so much time in along the, what you call this, the supply chain, and also we had to pay around 30 to 100% of the cost, both to legal rent seekers and also to corrupt ones. So it's, you know, if we could produce everything
that's the challenge, and that's why I would say, like, I mean, it's a struggle for us to get cartridges for Illumina at the moment, and like even clinical labs who are doing like life-saving testing are struggling, and so that is the problem everywhere. And so any kind of technique where you don't rely on those supply chains is definitely favorable over like going the MinION route, where you need flow cells from them, there's no other option, or you need certain reagents from them, there's no other option. And that's why I try and, as much as possible, steer clear of those kinds of things. I mean, for us, it's easy to get stuff from NEB and Thermo Fisher, for example, but I think the approach does need to change. especially because like if we learn anything from this pandemic, it's that it like shipping stuff on dry ice to South Africa is incredibly complicated. I think it cost us £500 to ship stuff on dry ice because there's like one company that does it because it's like a dangerous good and therefore only one company handles it, and so they just charge you as much money as possible to ship things.
Sounds familiar.
So I mean, yeah, no, exactly. This is the thing. It's like it is a familiar problem, and so it's the same around like production of standards and things like that. You want them to be made in place as much as possible, or like the reagents to be made in that place as much as possible. And I know there's quite an emphasis on trying to make it as cheap as possible to do it, or going like the secretory route and things like that. But I don't think the thing is like you could buy the protein purification equipment for one university in your country, and people could make the enzymes and sell them. Basically, have one university facility that produces enzymes for the entire country and uses the same equipment that we use here to do the same thing. I think that's a better way potentially of doing it than trying to go down the route of like everyone has to think of like how you can secrete the enzyme into the medium and then go that route for purification, because like one purification that you do in a university could be enough enzyme for the entire country for a year. So, and a lot of the stuff is off patent. I mean, the things like fusion and things like that, which are basically off patent, which are really good enzymes, but yeah. So I don't always think of this like as cheap as possible as I'll have a conversation with Jenny about it. Because some of the stuff she's doing is obviously around that, but it's like you need to be competing on the same playing field in the end, I think is the thing. Is like it doesn't make sense that you guys want to settle for reagents that are not as good as the ones that we can get a hold of. It's like you need to do the best, like you pick the best possible method, I suppose, even regardless of the cost, because you will recoup the cost by not having to pay NED or paying shipping or things like that in the long run. But anyway.
Yep.
Same sentiments.
Cool.
But I don't like the attitude, as I say, I don't like the attitude where it's like, oh, just because you're from a global south country, you should settle for something that's not as good as what all the other countries will get. Like, it doesn't make sense to me. It's like you should really be striving to use the same reagents.
You know, we'll take whatever we could get.
Yeah.
Cool. All right. Hey, thank you so much, Michael. This has been very interesting, and hopefully this will just be a start of like the exploration of these options for us, because we've seen how difficult it is to actually get hold of these reagents. So thanks once again.
Yeah, I'm quite happy to send them to you. So just give me a shipping address and I can send you the plasmids on like filter paper.
Cool.
If you're interested.
Yes, once we have the capability of producing these
well, I mean,
enzymes.
you have the capability of producing the plasmids at the very least. So you would have a kind of a control similar to a G block for that.
Cool. After this, we'll ask you like maybe some resources now for us to set it up.
Cool, no problem.
All right. Thank you so much.
Cool.
Have a
Thanks,
great day. Bye.
Thanks, you too.
Thank you. Very interesting talk.
Thank you.
Thank you. Bye.